Good morning, friends. Myself, Richa. I hope everyone is well. So, today, my important topic, what I'm going to start today is NER. So, let's start with this. NER. Right? So, first of all, I previously taught you about this NER. It's a widespread form of DNA repair that is known as NER, that is nucleotide excision repair. N stands for nucleotide, E stands for excision, R stands for repair. So in the previous slide, I showed you that NER, how it works in prokaryotes, in E. coli. Now I'm going to talk about how this NER repair system works in eukaryotes, like mammals, humans like us, or rodents, or yeast like this. So how it works basically, so you can see here the slide. So let's suppose UV radiation comes and the TT dimer has formed here, TT, right? So first of all, damage will be recognized by our body. So the TT dimer, either it will be removed by photolysis action with the help of photoreactivation, or it can be removed by NER in our body. So first of all, first protein which is helpful here in recognizing the damage is XPC. Next comes XPA. Next comes RPA. So these all are helicase activity. So next comes TF2H, which has two subunits, XPB and XPD. So here is the TF2H. So here comes all these things, this X. Now basically the damage is recognized, right? So here the DNA damage, that is thymine dimer, is recognized by XPC first, followed by binding of XPA, RPA and TF2H, right? Next, after this, what will happen? XPG will come here and XPF, or you can say ERCC1 proteins will come. You can see here this ERCC1 is here, XPF is here, XPG is here, and this blue one is XPC, yellow one is XPA, right? So these all are recognized by proteins. Basically, after that, the cleavage will be done and excision of oligonucleotide, that is 30 bases. The TT dimer will be removed, and next DNA Pol delta, RFC and pCNA proteins will come, and ligase will seal the gap here. Okay, so basically this nucleotide excision repair, these all proteins will work in conjunction in the case of eukaryotes like yeast, rodents, humans, right? So in CSIR NET exam, they can ask you like the series of proteins, like first of all, what here XPC, A, RPA, TF2H is working, and how much subunits TF2H is holding, like 2, XPB and XPD, right? And after that, these are replaced by XPG and XPF. This type of questions they can be asked. Or in the last one, you have to remember the DNA Pol delta will come here along with RFC and pCNA, and ligase will seal the gap. So they can ask you about the activities. Which one is the correct option in part C? Okay, so what happens, my question is, like if there is a defect in NER mechanism in our body, so what kind of diseases can happen? If there is defect in this DNA repair, which is normally occurring in our body, so we can suffer with xeroderma pigmentosum, that is XP. Second is cocaine syndrome. Third one is trichothiodystrophy. So first is xeroderma pigmentosum. It is a rare genetic disorder I must tell you that affects approximately 1 in 2 lakh people. Individuals with this disease are extremely sensitive to UV light and develop multiple skin cancers on their bodies that are exposed to sunlight. Okay, so this is XP, that is xeroderma pigmentosum. Next comes cocaine syndrome. It is a rare genetic disorder which is characterized by impaired development. So development will get impaired here. Or you can say premature aging and extreme sensitivity to sunlight. Extreme sensitivity to sunlight. Next comes trichothiodystrophy. Trichothiodystrophy, it is a rare inherited disorder which is characterized by brittle, sulfur deficient hair and a range of other symptoms including intellectual and growth impairment. Growth impairment. And photosensitivity and dry and scaly skin it results. So that's why these all our mechanisms are very important to function properly in our body. Otherwise, we can suffer with various diseases. So the next question which the CSIR can ask about this NER in mammalian cells, like if there is a defect in NER, how many diseases can occur? So these three you have to remember: XP, cocaine, and trichothiodystrophy. These three kind of defects can occur. So I taught you about the two common questions what they can ask. First, of the series of proteins. First, how the damage will be recognized, which activity they are doing, that is helicase activity. Second, the proteins which are coming in the first part, how they are replaced in the second part. This question can come. And after that, finally which enzyme action will work, this they can ask in the series. And next, defects like this. So these are the main two questions which can come from the this part of NER. So I hope everything is clear. All the best for your exams. It is mostly commonly occurs twice in a year in December. We are preparing, right? I hope so you can solve the questions on the daily basis, right? So thank you. If you like my channel, please like, comment on my channel, subscribe it now. My channel name is Richa1. Handle is @richa h3k. So this is my logo. If any queries regarding questions, please free to contact me at my mail ID richa82304 @gmail.com. Thank you. Have a nice day.